past winter and spring. This was a new idea that got thought up sort of this year, although it's been in the works for a while. It's been a really, really fun webinar series. We are so appreciative of all the people that have come out to these webinars and the presenters. We've learned a lot. We've had anywhere from, I'd say, a minimum of 30 participants to over almost 100 at some of ours. So it's really cool the different people that we've been able to reach. And so, like I said, thank you so much for being here today. And I'm going to hand it off to Charlene. And just one more little piece of housekeeping, I guess, before we start is if you have any questions, please submit them in the chat, and then we'll go through the questions at the end. And of course, at the end, you're also can totally just ask your questions out loud too.
All right, well, I'd like to introduce Mark Ward. He's an ecological consultant out of Bristol, Maine. He's a graduate of the Field Naturalists Master Program at the University of Vermont. And that's where he really got into invertebrates when he took field zoology from Ross Bell, who is was a well-known ground beetle specialist. He's consulting, his consulting has ranged from ecological field studies to conservation planning projects, and has done a number of insect-related projects for both the state and local governments, land trusts, and private landowners. And he's been working on the ringed Boghaunter in Maine since 2001. And that's what he's going to be talking about tonight. So, Mark, it's all yours.
Okay, great. Well, let me start by I'm gonna try to share my screen here, but I'll start by saying that I am really happy to be here. I've attended the other, or at least some of the other sessions, and found them to be really fascinating. So I'm really excited to be here with you. And can people see my screen?
Yes.
Okay, great, good. Yeah, I mean, this is a species, the ringed Boghaunter, that I was fortunate enough to have the opportunity to get introduced to back in 2001. And the story that I'm going to relate to you, I think, is one about a very fascinating creature. And I hope that you find this creature to be as fascinating as I do. And it has a lot of things to reveal, and we're still only scratching the surface of it. So the title of the talk is, of course, The Ringed Boghaunter, Challenges and Strategies to Detect and Protect a Rare Dragonfly in Maine. So without further ado, let me begin, and hopefully we can make this work. So let's start by doing a little background on the ringed Boghaunter. First of all, it's a member of the emerald family, which is the Cordulidae, sort of the forgotten family of dragonfly species out there. I think of, everybody thinks about the darners because they're so big and sexy, and then the libellulids, the skimmers, who you see at every pond. These are the cordulids are the group that people often forget about or don't pay as much attention to or don't see as often. This species was first discovered in the 1870s in New York State by Joseph Lintner. I'm sorry, is that a question? So, and it was later described in 1878 by Hermann Hagen, who was a German entomologist. It was elevated to the level of genus in 1913 by Edward Williamson. And it was, in fact, the last North American, or at least the US and Canada, Cordulid genus to reach that status. And its larva wasn't described until 1970, when a paper came out describing it. Up until that time, it had been sort of mysteriously eluding people. And that's part of the story of this species in general, that it's elusive. So very few of you probably realize that the ringed boghunter is also one of the only dragonflies that's known to sometimes have two heads. And so for those of you out there who are scratching your heads, yes, this is indeed an April Fool's. So no, the ringed boghunter dragonfly does not have two heads. But I thought, you know, hey, it's April 1st. For those of you who might see this in a recorded fashion, it is April 1st. And so I thought, why not have a little fun? So seriously, everything that I say from here on out should be at least somewhat true. So the ringed boghunter is interesting for a variety of reasons, and one of which is because it's globally rare. It's ranked G3, which puts it as being vulnerable, and part of the reason for that global rarity is an extremely limited distribution. On the left, you can see a range map that's adapted from Dunkel's Dragonflies Through Binoculars. Originally, we thought that this species was really only found in the Northeast, from the Atlantic states up to, say, Massachusetts or so. But in the last few years, we found that it's in states in northern New England and also in Michigan and Wisconsin. Those were discovered. The species was discovered in those states in the late 1990s. So we've sort of expanded its range of what we know about it, and we have more populations. We're aware of more populations than we were previously, but nevertheless, it's of concern because there is evidence of its extirpation. I talked about the site where Joseph Lintner first found this thing in New York State in the 1870s. No one's ever been able to find it since in New York. So it's believed to be extirpated in New York. The population in New Jersey is also of questionable status. And so there is some evidence that this species is subject to decline in parts of its range, at least, even though we have found it to be present in other places since then. So let's talk a little bit about its general biology. I'll start with the breeding habitat, which is characterized by open, acidic wetlands with standing water and so-called soupy sphagnum. So generally, we're talking peatlands, but maybe not your classic boggy peatland, but a more of a fenny, acidic wetland that you can walk through, at least part of. And when you're walking through it, you find that your waders are getting glommed on with sphagnum that's floating in the water column. This seems to be characteristic of the breeding habitat of the ringed boghunter. So it's a relatively, I wouldn't say an uncommon habitat that it's found in, but it's one that is at least unusual. Now, in these breeding habitats, there also has to be some sort of emergent substrate present. This is to allow the species to find a place to emerge from the aquatic habitat as it transforms from its larval stage into an adult. And also, one of the things that we know about this species is that it tends to favor habitats where the hydro period is one that limits fish presence. So these can be ephemeral wetlands, wetlands that dry out most years, some years, or relatively infrequently. But in one way or another, it can be isolated wetlands that don't have clear outlets, and that can limit fish presence. But the absence of fish seems to be something that allows this species to compete relatively well in these types of breeding habitats. Some fascinating other characteristics about this species is that it has an extremely early flight season. It's essentially the first dragonfly, other than the green darner, maybe, the migratory Anax junius. It's essentially the first resident species to emerge from its wetland in Maine. And that, I'm going to talk more about that shortly. It also has an extremely brief flight season. So it is on the wing as an adult for anywhere from 2 to 3 weeks, maybe a month at the most. But it comes out really early, say, early May, and by the beginning of June, it's gone. As an adult, at least. You don't see it as an adult anymore after that. So it has a very sort of short window where you can find it flying as an adult. And so another characteristic that's interesting about these things is that the adults don't generally hang out at the breeding wetlands. So once they emerge from the wetlands, they tend to fly off into the surrounding upland areas, wooded areas typically, and they hang out relatively surreptitiously in those uplands. So they can be quite difficult to find for all of the reasons that I've laid out as adults. They fly really early when most people aren't really looking for dragonflies. They're not flying for very long, and they occupy these sort of upland habitats that you might not know are places where they would be because they're not around the breeding wetlands very, very often. One of the interesting characteristics that we really don't know much about is that we're not really sure whether these things have a 1 year or a 2 year life cycle. In other words, do they complete their full life cycle? Do they lay their eggs in the late spring and then have the next larva emerge and transform into an adult the following spring? Or can that take 2 years? There's some evidence to suggest that maybe it does a little bit of both, depending on the specific characteristics of the wetland, but we really don't know whether it has a 1 or 2 year life cycle. So all pretty interesting characteristics, I think, for as far as dragonflies go. So let's talk a little bit about the history of survey work for this species in Maine. Its presence was predicted back in 1988 by Dale Schweitzer in some work that he was doing for the Nature Conservancy. It saw limited survey effort in the early 90s. John Albright, who I also worked for the Nature Conservancy, and some folks from IFNW put in some, from the Fish and Wildlife, State Fish and Wildlife, put in effort in the early 90s looking for this thing, but it was a little haphazard, a little random. It was eventually discovered in 1995 by Mark McCullough and Jamie Haskins in southern Maine at a population in South Berwick. That was the first known occurrence of the species in Maine and warranted a publication in the Northeastern Naturalist. There was a focused study of that metapopulation in South Berwick in 1997 by Karen Hopkins, which revealed a lot of information. And then we've done strategic surveying effort from 1998 to 2013 that the Department of Inland Fisheries and Wildlife did. So there were a bunch of people who participated in that over those years. Then there was a little bit of a hiatus where there wasn't a lot of work done. And then the last couple of years, Philip de Mainadier and myself have sort of resuscitated our effort to look for this species and understand its habitats and its populations. So what are some of the objectives of doing this field work when we go out looking for this thing? We're trying to determine how rare it really is in Maine. Is it rare because we finding it can be so difficult, or is it rare because it truly isn't there? And part of that is to identify breeding wetlands, identify habitat essentially where this thing breeds successfully so that we can ultimately try to protect it. And we're also trying to understand the upland habitat use. I mentioned that the adults leave the breeding wetlands essentially right after they emerge, and we're trying to understand how much upland habitat they use, what kinds of upland habitat they are most likely to be found in, etc. So surveying methodology, in other words, how do you find a needle in a haystack? This critter is not the easiest thing to find. So one of the things that we try to do is we want to make sure that we know when they're out. How do we know that they're out yet in a given year when we go to survey? We don't want to spend a lot of time before or after they've already flown as adults. We also want to know which wetlands to survey in, because the time period during which these things are on the wing is so brief. We want to maximize our ability to find this thing, and so we want to highlight the wetlands that we work in to maximize our effort during that brief window of opportunity. And then we also want to know how much time should we spend at a site? How much time, is it worth to spend at a site? So those are sort of some of the questions that frame the way that we've shaped our efforts to look for this thing. So how do we know that they're out? Essentially, we document the dates of emergence at known population sites. So we go to 2 reference sites that we know of, and we look for when this thing is coming out at those 2 sites. And the nice thing about that is they're 2 very different and a dirt road that goes through it, where oftentimes we find the adults. And the other is an isolated wetland where we essentially look for the species emerging in the wetland proper. And these are some data that we have for those 2 sites over the last 15, 20 years. The main sort of thing to glean from this is that the sort of average time that these things emerge is in the first week in May, but the range can be from essentially mid-April to mid-May, when we first see them on the wing. Here's a visual representation of that same data. This shows 1995 to 2013. And because it's a relatively long data set, you can see a pattern that's emerging here where, in the early years, in the 90s, the period during which we were finding this species for the very first time of the year was relatively late in May. On the left, that axis is a little difficult to interpret. It's the number of days from April 1st. So essentially the 30 represents the last day in April, 35 would be May 5th, 40 would be May 10th on the left-hand side. And then the 2 colors represent the 2 sites that we've monitored for this. So we looked at these data and we said, we know that the climate is changing. We know that spring is coming earlier and earlier. It looks like there's a really nice pattern here to demonstrate that this species is emerging earlier than it was in the late 90s. Even over that relatively short period of time, we seem to be seeing this. So, like I said, there was not a lot of work done on this species between 2014 and 2018, but we have looked at it in the last couple of years, and lo and behold, in the last couple of years, the pattern doesn't hold up so well. So we might need to continue to look at this and over time to see whether the pattern of seemingly earlier emergence dates holds up through time. Which wetlands to choose for survey work? This can be challenging. And so what we've done is, some of it is systematic, some of it is based on sort of less systematic effort. So one of the things that we do is we follow up on leads. Essentially, if somebody who's aware of this critter sees a wetland that they feel like looks like good habitat at any time during the year, we try to collect those possible locations and try to visit them the following year if we can. We also have used national wetland inventory maps to target certain types of categories on those maps. One is the broadleaf evergreen category, which typically is characteristic of leatherleaf, and another is the category, the sort of A modifier category, which is characteristic of acidic wetlands. And then partially by chance, as when we're out there, sometimes we see things that we might not have thought looked good from the maps or that we weren't aware about on leads, but in going to other places, sometimes we see wetlands that turn out to be reasonable to survey for this species. So this sort of summarizes the known wetlands that we found in Maine by National Wetland Inventory type. I wouldn't get too hung up on this. It sort of breaks things down. I don't know if you can see my cursor or not, but if you look at the scrub shrub category across the top, a lot of the wetlands have at least some portion of the wetland falls into that scrub shrub type of habitat. There's some other things that maybe we can learn from this, but it's sort of broad and general and doesn't really tell us a ton, unfortunately. How much time do we spend at a site? Well, that depends. And in order to figure out how much time is worthwhile to spend at a site, we've developed a scoring system for the wetland. In other words, a system that tells us, like, is it a good wetland that we should bother spending time in, or is it not a very good wetland based on what we know about the habitat needs of this species? So the Habitat Potential Index, or HPI, is basically a system that we've devised to score wetlands based on the presence or relative abundance of, is the canopy open? These wetlands where we find this species seem to be places where there's a fair amount of light that gets through. If it's a really small wetland and on the south side of the wetland there's dense hemlock or something like that, it's probably not going to be a great ringed boghaunter wetland. Or if it's a forested swamp that has a lot of standing trees in the swamp, same thing. Sphagnum, like I mentioned earlier, especially the suspended or soupy sphagnum, but also sphagnum that's anchored to the bottom that you can see through the water column that's present. We score the wetlands based on the presence of those things. And then, does it have emergent substrate for the larvae to come out and emerge? Other considerations that we take into account are the water depth and the suspected, sort of the estimated hydro period of the wetland. Does it fall within that sort of ephemeral or semi-permanent category where it might dry up in certain years? Those are all things that would be potentially positives. So surveying techniques, we tend to spend more time in and around wetlands that have high values, ones that score well on our index system. And when we're looking for these things, we search for adults, typically in the uplands around the wetland. And then we also search for the exuviae, so the shed skins of the larvae in the wetland proper, and also potentially for emerging adults, emerging teneral adults that may be coming out. So when we're looking for evidence of this species, the holy grail really is to find an emerging teneral and an associated exuvia. Doesn't happen that often, but when it does, it basically convinces us that, yes, this species is here and it's breeding in this specific wetland, and that's great. That's exactly what we want to find. It doesn't happen that easily. So instead, we also have to look for adults. And typically our first sort of lead in an area is an adult observation in an upland. So one or the other of us might find an adult, and then once we've found an adult, then we start narrowing our search for wetlands in the vicinity that this adult might have come from. So adults, I'll just go on. Adults oftentimes are found like in places that are sort of open, so they might be basking on a rock or on a dirt road, but it would be near, obviously, a breeding wetland of some sort. The other thing that we look for are exuvia in the wetland. So exuvia that are not associated with a teneral, but exuvia that were left behind where the adult has emerged and left, and all we find is the shed skin of that larva. And that happens, I would say, of all of these types, that's the most common thing that we find is exuvia in a wetland, Williamsonia exuvia. So how do we know when we find an exuvia that it actually belongs to this genus of Williamsonia? Well, there's some characteristics that help us, and that is the dorsal hooks on the back of the or on the dorsal side of the abdomen of the larva. This this genus has dorsal hooks on abdominal segments 3 through 9, and that's unique for the genus. And then also, it only has a lateral spine on the last abdominal segment, on the 9th abdominal segment. Most of the other cordulid species have have lateral spines on the 8th and sometimes the 7th and even earlier spines, or earlier abdominal segments as well. So again, we're looking for the 9th lateral spine only, and then dorsal hooks on the 3rd through 9th segments. And once we have that, then we know that we're looking at a Williamsonia exuvia. So that's great. That tells us a lot of information. So this is something that we came across early on from a report from Virginia Carpenter in Rhode Island, that basically searching for exuvia is the most reliable, conservative, and economical method of monitoring Williamsonia lintneri, or the ringed boghaunter. So that's great, right? Because I said exuvia are the easiest thing of the 3 to find. So if you can find exuvia, you're all set, right? Well, unfortunately, in Maine, it's not quite that simple. And that's because we have another species that is also found in Maine that belongs to this genus, whereas in Rhode Island, they only have one of the 2 species of the genus. So in Rhode Island, you can use the exuvial search as your primary method to find the species. In Maine and other parts of its range, not so easy. So these are range maps of the 2 species of Williamsonia, and you can see that their range partially overlaps. The ebony boghaunter has a broader distribution than the ringed boghaunter does. It's not nearly as rare as the ringed boghaunter is, and certainly not as rare in Maine. So let me give you a little background about the ebony boghaunter, this other critter in our midst here. And it was also first discovered in the 1870s in Winnipeg, although it wasn't identified as such until 1923, when it was described by Edward Williamson. So this was later than the ringed boghaunter description, even though it was found at the same time. For a long time, the 2 species were thought to be the same species. And you can understand that, especially back in that time period when they didn't have iNaturalist and other ways to post photos of specimens. These folks were working, oftentimes it was European entomologists who did descriptions of some of these things. So, getting a specimen to Europe, it happened. They mailed things back in those days, but things didn't happen as efficiently as they do now. So, and the larva for this species wasn't described until 1993 in a paper by Charlton and Canning. So the larva of the ebony boghaunter was one of those things that I think it was Walker who had put out a challenge to his students to find the larva of this thing for many, many, many years, and it wasn't actually found or described until the early 90s. So that's pretty recent if you think about it. I mean, in the grand scheme of things. So so again, ebony boghaunter in Maine, much more broadly distributed than the ringed boghaunter is. It's not a rare species in Maine. It's an uncommon species, but it's not rare. So it's not state threatened or state endangered or anything like that. In fact, the Maine damselfly and dragonfly survey, all the red blocks in this map that you see are examples of records for this species that were identified as a function of the Maine damselfly and dragonfly survey project in the early 2000s. So we really expanded our knowledge about its range in Maine. And essentially it's found statewide, whereas the ringed boghaunter, on the other hand, has a extremely limited range, at least as far as we know, in Maine, essentially limited to York County and the very southern part of Oxford County in the sort of Brownfield, Freiburg area, which is sort of a weird little it's a weird little corner of Maine. You know, it's sort of, in some ways, it's southern Maine like. It heats up a lot quicker than some of the coastal areas in southern Maine, and so it has a really interesting, sort of unusual local climate factors. So if you do find the adults of these, the fortunate thing is that the adults of these things are, if you have them in hand or in your camera, are relatively easy to tell apart from one another. So the ringed boghaunter has these orange or sort of light brown, pale abdominal rings all along its back, whereas the ebony boghaunter only has white or cream colored bands on its first or second abdominal segments. So it doesn't have the rings or dorsal lines across its abdominal segments along the rest of its body. So that's really helpful in terms of being able to distinguish the adults from one another. Another challenge with these species is that their flight times largely overlap. The ebony boghaunter also is an early species, not quite as early as the ringed boghaunter is, but still very early. It can be found. This is actually an old table. I'm not sure when. Anyways, their flight times largely overlap, although the ringed boghaunter tends to come out a little bit early, maybe a week or so before the ebony boghaunter does. So on top of that, their breeding habitats also overlap. So with the ebony boghaunter having a slightly more liberal perspective of breeding habitats. In other words, it's more able to tolerate a broader range of different types of peatland habitats than the ringed boghaunter or Williamsonia lintneri is. So again, these are all things that make these two species similar and somewhat difficult to distinguish from one another. On top of that, the exuvial characters that distinguish the species can be challenging. So when the folks described the larva of Williamsonia fletcheri, the ebony boghaunter, in 1993, Charlton and Cannings, they pointed to several characters that can be used to distinguish between the two species. And these focused essentially on the length of the lateral spine on the 9th segment and the length of the dorsal hooks on the 8th and 9th abdominal segments. The challenge is that they were using a very limited sample size. They only had 10 of one of the species and 6 of the other species. They had a very small sample size on which to base these characters. So moving forward, we've since measured a whole bunch of these exuvia for specimens that we know were associated with a teneral adult, so that where we were able to confirm that the individual that the exuvia that we found was truly associated with a recently emerged teneral adult. So we borrowed specimens from different folks in different states and provinces. We've measured a lot more than Charlton and Cannings did. And the unfortunate conclusion that we reached is that no single morphological character is 100% reliable to distinguish between the two species. So in other words, those dorsal hooks, the lateral spine lengths, all of the associated measurements around the lateral spine, none of them is a reliable metric to use. So we can't go in measure those things and say, aha, this exuvia, which was not associated with a teneral adult, belongs to species A or species B. It does seem to be true that the general pattern that they talked about, where there's slightly longer lateral spine in one species than the other, slightly shorter dorsal hooks in one species than the other, that does tend to be true, but the ranges of measured things that we've identified with associated exuvia, unfortunately, all overlap. So we can't rely on that as a metric to determine that an exuvia that we found belongs to species A or species B. Okay, so what does that mean? That poses a whole set of challenges then for what it means to determine whether a wetland is indeed a breeding wetland for this species. And so as a result, we've developed what we call an evidence-based categorization of breeding wetlands using the information that we have. So basically, a wetland is considered confirmed if we have if we observe an emerging teneral and its associated exuvia in that wetland. And we have 11 of these wetlands in Maine that we've confirmed by identifying an emerging teneral and its associated exuvia. A probable wetland is one where we found an adult ringed boghaunter in the nearby upland, and at least one of these unidentified or unassociated Williamsonia exuvia in the wetland. Or we saw an ovipositing female in the wetland of Williamsonia lintneri, or an observation of a mating pair in the wetland or on the shoreline, or an observation of a teneral adult in the wetland or on the shoreline. So we have 5 of these wetlands that we are in this probable category. We're pretty sure that they're breeding wetlands, but we don't have that last sort of holy grail of the associated teneral and associated exuvia. And then finally, we have a bunch of wetlands that we consider possible wetlands, ones where we found Williamsonia exuvia, but we don't have any sort of other observation that points to it being Williamsonia lintneri. So it's essentially what would pass in Rhode Island as a confirmed wetland in Maine. With only the exuvia, we have what we consider a possible wetland. And the first 2 of these categories, a confirmed wetland and a probable wetland, are ones that actually have some sort of regulatory authority for this species, whereas possible wetlands essentially do not. Oops, sorry, there's one other category that falls under the possible, and that's if we have an adult ringed boghaunter near a wetland with a high index value, but no exuvia found. Actually, I don't think we actually have any of those, but it's one of our categories that we would consider to be the case. So, okay, so let's, I'm sort of using up my time here pretty quickly. So I'm gonna go through pretty quickly here some of the results of our breeding wetlands. We've surveyed more than 400 wetlands in southern Maine, and that's more than 800 site visits. 73 of those wetlands had high HPI values, so 18% of the wetlands have sort of the characteristics that suggest that it would be a good ringed boghaunter wetland, but only 25 of those wetlands have shown evidence of potential breeding. So those would be either confirmed, probable, or possible wetlands in what I just described. And only 16 of those, or 4%, are actually confirmed or probable wetlands. The estimated number of distinct populations for the ringed boghaunter that we have in Maine is 9. So 9 distinct populations, because some of our wetlands are sort of in very close proximity to one another. So we don't think of them as distinct populations. They constitute more of a meta population where it's essentially one population with multiple breeding wetlands nearby. Thinking about the adults, we've documented the flight times of the ringed boghaunter. In Maine, that's known to occur between April 16th and June 11th. But the vast majority of our observances of adults is in the month of May. We've also looked at distances to presumed wetlands of origin. So distances that the adults have traveled. We're interested in this because we want to understand how much upland habitat these things use. And so that range is anywhere from 300 to 500 or so meters. And for isolated wetlands, wetlands that we know only have essentially one wetland associated with them, if we take the median value, the median value for the distance that we find is around 62 meters. We know from other studies that these things can travel hundreds of meters from their wetlands in other states. In Rhode Island, for instance, they have documented that. But in Maine, our ability to do that with adults has been very limited. So I'm not sure. I think I'm gonna sort of whip through this really quickly because I don't want to spend too much time. We've looked at the exuvial microhabitats, where the exuvia are found. We find that the water depth range for these things is anywhere from 9 to 71 centimeters in depth beneath where an exuvia has come out associated with a tenorel. And we tend to find that there's lots of this sphagnum, both anchored and suspended in the 1 meter square proximity around where we find these exuvia. And another interesting thing that we found is that most exuvia that we found have been in the northeast quadrant of a wetland. Now, this is a schematic of that sort of shows that these things seem to be heavily skewed towards the northern and or western part of the wetland. But I would caution us reading too much into this because most of these exuvia that we found of associated exuvia that were associated with a tenorel adult are from a single wetland. So I don't know that this is necessary. This could be characteristic of that particular wetland, but not necessarily a characteristic of the species in general. Let's see. I'm going to sort of skip over. Well, let me just say that this table demonstrates sort of how much time it's taken us to go from an initial observation of an adult at a spot until we know something more about the population, the breeding status of the wetlands associated with that population. And so this last column on the far right basically tells you how many years it took us from the time we first saw an adult in the vicinity until we were able to document one or more of those wetlands as a breeding wetland. And so it can take it's taken us up to 12 years in some cases to confirm that a wetland was a breeding wetland. Although more often it's with anywhere in the 0 to 3 range. So 0 to 10 years within a population, 0 to 12 for an individual wetland. But most cases, when we find an adult within the first 1 to 2 years, sometimes that year, sometimes within the first 1 or 2 or 3 years, we're able to confirm that we've got a breeding wetland nearby using that evidence-based system. So to conclude, Williamsonia lintneri, the ringed boghaunter, remains rare in Maine. It's a state-threatened species, so it's still quite uncommon. When I started this work back in 2001, it was actually a state endangered species. So by finding some of the new populations that we have, we've actually sort of downlisted it from endangered to threatened. If I had to guess, I would guess that there are more populations out there that we still have not detected. And it is a species of greatest conservation need, a priority 1 species in the state of Maine's State Wildlife Action Plan. The Habitat Potential Index has helped us as a tool to help fine-tune wetlands that we think might have the potential for breeding habitat. The presence of the ebony boghaunter complicates the task in Maine and in other parts of its range where both species are found, which we know there are several states where that's true. And the evidence-based categorization of breeding wetlands is essential for us to really help nail down whether a wetland is indeed a breeding wetland for this species in Maine, and to allow that wetland to have some sort of regulatory authority under the State Endangered Species Act. So just briefly, threats to this species in Maine, obviously the habitat loss from development pressures in southern Maine. Southern Maine is notoriously the part of the state that sees the greatest amount of development pressure. Habitat degradation can be a problem, even if the wetland isn't destroyed. The surrounding uplands can be sort of developed and hit pretty hard. And one of the potential challenges is compromised water quality in these wetlands that can make it difficult for this species to survive. So regulatory ramifications for this species, all 16 of the confirmed and probable wetlands and the associated uplands with those wetlands trigger regulatory review for this species. And many of these wetlands are considered significant vernal pools. So that, because many of them are isolated wetlands, this species, because it is a species that's associated with vernal pool habitat, in addition to or instead of amphibian counts in significant vernal pools, this species' presence as a breeding wetland can constitute its protection as significant wildlife habitat under the state's significant vernal pool law. And then that goes, one of the things that go along with it are 250 feet of the surrounding upland are also protected as a result of that. So conservation status of the populations that we're aware of in Maine, at least 3 of the 9 populations have some conservation protection. And the data that we've been able to accumulate about populations in the state are provided to the beginning with Habitat Program for conservation planning purposes. Future directions, we'd like to really develop a more reliable method to distinguish between unassociated Williamsonia exuviae. That sort of effort is ongoing. I won't go into it now, but we have some ideas about that. We want to hone our understanding of the triggers for the emergence time and continue to track the potential link to climate change to see whether that pattern is indeed something that manifests in the future. And we want to explore techniques for tracking this species in the upland, in its upland habitat movements. We've looked into some different techniques to do that, but it's a very small species and very lightweight. And so any of the sort of tagging things that other people have done, it doesn't seem to be robust enough to allow for that, at least at this point in time. And we want to expand our awareness of this elusive species and develop a knowledge base of local observers. So for yourselves, we're coming up on ringed Boghaunter time. Those of you in the Maine Entomological Society especially who live in York County or Oxford County or even Cumberland County, I would say beginning at the end of April and into May, be on the lookout for this species. Because if we can find adults of this species, that will more likely than not help us hone in on its breeding wetlands nearby. So finally, to wrap up, I'd like to acknowledge that the funds for the work that myself and essentially all the other IF&W folks who have contributed to this effort have come from Maine's Non-Game and Endangered Wildlife Fund, proceeds from the loon license plate and the chickadee checkoff on your tax forms, and from the U.S. Fish and Wildlife Service endangered species grants to states, the Section 6 money that they get for the state endangered species. I'd also like to thank my colleague and mentor, Philip de Maynadier, who is a consummate naturalist. I always learn, he trained me to look for this thing back in 2001. And every time I'm out in the field with him, I'm constantly impressed at the things that I learn from him, the things that he sees and hears. And it's great to go out in the field with him and see through his eyes for a day. Lots of collaborators who've helped in a variety of ways from different states and from here in Maine to provide us with specimens that we've looked at and other helpful tools and images that I borrowed for this program from the following sources. And with that, I will take some questions.
Great, thank you so much. That was wonderful. We've got a lot of questions. So
oh God, okay, yeah.
I'll ask a few of these ones that are, well, I'm going to ask, I guess, all the ones that are in the chat, but if anybody wanted to ask a question in person, just, you can kind of just pop open.
Put that thing down.
All right, so we're going all the way back to 7:07 p.m. Oh, we got a visitor over here. This is for Annie. All right. So Charlene asked, could it have moved to the Midwest due to people selling dragonflies for mosquito control?
I doubt that. I doubt that because I don't think that they're taking the mosquitoes for mosquito control from the kinds of wetlands that this species would be found in. So
Okay.
I can't say it's impossible, but I would say it's highly unlikely.
Okay, great, cool. All right, Kathy asked, is anyone looking for it in Canada?
I believe, yes. I mean, certainly, I know Brian Pfeiffer in Vermont, I know that's not Canada, has been searching for this species over there because it's sort of a gap there. If you go to the NatureServe website, it suggests that there's a record in Ontario for this species, but I don't know, I'm not sure about that. I've never seen evidence of it. So somebody might know more than me about that. I think I wouldn't be entirely shocked if even in Nova Scotia they were to turn this thing. Those guys out there are pretty good naturalists. They may or may not be looking for it. I don't know, but it wouldn't be. I mean, that's a little bit of a stretch, but it's possible. Certainly that part of Ontario that dips down by Detroit is probably the best possible habitat for this thing in Canada, I think.
Great, thank you. And I know you did kind of talk about this some in the presentation, but somebody did ask how difficult the identification is, which I'm gonna tie in my own question on. Is there any field guide that we could use if we were going to be looking out in the field for this?
For this species? Sure. It's let's see, what do I use? What's the best resource? I mean, let's see, what do I use? The Dragonflies of Massachusetts is probably the best resource. It's Jennifer Luce and it's 3 authors. She's one of them. Matt, I think Blair Nicola is one of them, and Matt. Can't think of the third person's name. I apologize, but yeah, Dragonflies of Massachusetts has most of the species of dragonfly and damselfly that we have in Maine are found within that book. Most, but it includes these 2 species for sure. So that's a good resource. They have good information in it, good pictures.
Perfect. All right. Chris said, Mark, I wonder if remote sensing might catch the soupy sphagnum type wetland from satellite imagery. Do you know if that's ever been tried to identify potential habitat?
I don't, and that's a really good idea. I would love to know more about that because, yeah, remote sensing. I don't know what that means exactly. Are we talking like LiDAR or something else? Or I'm probably not LiDAR because I think that does but yeah, if there was a tool that would detect that, I would love to know about it because that would really help us a lot because that's probably the most important characteristic that we're looking for when we're out there to zero in on potential habitats for these things. So it's a good idea.
And then Deb asks, does DNA testing confirm that the ebony and ringed bog hunters are different species?
Let's see, does DNA evidence confirm? I don't know that anybody has done DNA to confirm, but morphologically, the adults don't look anything like each other, and they're so and I think that they're sort of true to that. So I don't think that there's at this point in time, I was talking about like back in the late 1800s and early 1900s when there was sort of some confusion about them. I don't think anybody thinks that there's confusion about them as being distinct species.
I usually, and I toss in that I'm pretty sure when it comes to dragonflies, I think there's some pretty good keys on the male genitalia. So
yes, the genitalia are different from one another. There's other characteristics besides the male and female genitalia are different from one another on these two species. So, great, and those are in the descriptions of the species as well. So.
Awesome. There's quite a few more questions, but I'm going to pause reading from the chat and just give anybody the opportunity to ask an in-person question or bounce off of anything.
I was wondering if this is related to Chris Shorn's question, whether there's been any correlation noticed between breeding locations and the species of sphagnums found there.
Yes, it's a good question. Julie Lundgren back in the 90s did some characterization of sphagnum at these wetlands, and there were two species in particular that she identified as potential indicator species for indicator sphagnum species for the ringed Boghaunter species. Sphagnum phalax is one, and Sphagnum cuspidatum, I think, is the other. And I have to admit that I am not that good at my sphagnum identification. And it's one of those things that like every year I tell myself, yeah, this is the year I'm gonna learn how to identify the sphagnum to the species level. And yeah, I mean, the challenge with that is like you have to be good enough to do whatever it is that she did that was able to help her identify to recognize those species. And I don't know if that involves, I know with sphagnum, oftentimes it does involve at least familiarizing yourself with them using a microscope. Now you might get to the point where you're able to identify them to the species without the microscope in the field in the future, but if you have to take the specimens home and, in other words, if you don't know in the field what species it is, it doesn't help you that much unless you have to take it home and then identify it and then, okay, next year I'll come back, or, a few days from now, so yes, and I think it has potential to offer a good tool to somebody who's knowledgeable enough to use it as a field tool to really help you zero in on these things. So that was a long answer to a relatively simple question.
Thank you.
Yeah.
I've got a question. This is Roger. If these dragonfly inhabit vernal pools, then I assume that the larvae are resistant to dehydration.
Yes, and I meant to mention that. Yeah. Yes, it's well documented that they can withstand at least some level of desiccation. We don't really know. I don't think anybody's really like studied it in depth, but I know some of the wetlands that I work at dry out over the summer, at least dry out to my eye, So at the bottom of these wetlands in the late summer, if especially in a dry year, right? What you have is this sort of mat of sphagnum that was floating that is now like congealed on the bottom of the sort of mucky wetland. And I think that can act as an area that stays moist, even though it's, essentially the wetland is dry. There's no standing water in it. But that layer of sphagnum that's now fallen to the bottom of this pool on the underside of it can continue to have a level of moisture that seems to allow this species to withstand a certain level of desiccation. Now, at some level, of a threshold, does it get dry enough where the thing might not be able to survive? Maybe, I don't know, but it definitely seems to be able to withstand a relatively high degree of desiccation, I would say. So.
Thank you.
Very cool.
I know it's a really cool species. There's all these things about it. It's really interesting. Yeah. So.
We have a lot of questions from Hanson, but I just think one that he or she, I'm not sure, has a sort of a related question to that was, why in particular did you choose to study the ringed Boghaunter? Does it provide anything unique to the environment that other odonates do not?
I didn't really choose it. It chose me. That's what I would say. I mean, I sort of fell into the opportunity back in the early 2000s, and I have continued to be fascinated by it through this time, and to continue to have the opportunity to do work for IFNW on it, which is I am grateful for. What does it do? That's one of those questions that is, dragonflies in general, we know they eat a lot of the so-called pest insect species. So I'm sure ringed boghunters, like others, do that when they're flying around. And in the larval stage in these wetlands where I know mosquito larvae hang out. So if you're looking for a human benefit, that's probably the best that I'm aware of. I tend to not necessarily operate on those terms. My mother-in-law is a famous person to ask me, well, what does it do for us? And my answer is it doesn't need to do anything for us. Our knowledge that it exists and that our actions are potentially harming its ability to survive is enough for me to know that we should act on that in a way that helps protect it to the extent we can. So.
Great. Well, I just want to point out, 8:03. Are you okay to keep answering?
I am totally okay.
Cool.
I went longer than I thought in the talking, so yeah.
Cool. All right. Kathy asks, to what extent do the ringed versus ebony occupy different niches in the ecosystem besides flight time? Is it inevitable that one will take over the other? Which I know is related to the other question, but I like that wording.
Yeah. Like I said, in one of those slides, the habitat sort of preferences, I would say, for the ebony boghaunter are broader. It's just able to use a wider range of these sort of peatland habitats than the ringed boghaunter does. That said, we know that they can be found at the same sites using the same breeding wetlands. So we know that there are wetlands where both of them are present because we've seen both of them emerging from them. So I don't know if that answers the question entirely, but yeah, is that what was getting at?
I think so.
Okay.
Kathy, you can chime in if it wasn't.
Well,
yeah.
I was just
Go ahead.
thinking, we do see species becoming less and less common because some other species is more successful.
Yeah.
So I just wondered if part of what we're seeing is something that has nothing to do with people. I mean, there's obviously both of them can be impacted by degradation of the environment, but is something happening there where a generalist like the ebony is just inevitably going to push out this niche, the one that's more of a niche species?
I don't know. Obviously, I don't know, but I don't think that that's the most likely pressure on on the ringed boghaunter. There are other species in these wetlands too, ones that aren't Williamsonia, right? The Leucorinia genus. There's several species of Leucorinia, the white faces. One that comes out almost as early as the ringed boghaunter, the Hudsonian whiteface, and that thing can be there can be tons of those in these wetlands, much more abundant than than the ebony boghaunter is at a site. So in terms of crowd, I don't know. I'm not sure about what happens. I think within those wetlands themselves, the aquatic phase, the larval stage, which of course is the sort of the bulk of this species' life stage. They probably carve out their own little niches within those sort of aquatic microhabitats that allows them to cohabitate there. So
Great. Tom Hayward asks, are vernal pools acidic enough for this species?
acidic enough? Is that what the question was? Are
Mhm.
vernal pools acidic enough? I vernal pools is a really, really, really broad category. So there's lots of varieties of vernal pools. The ones in which this species are found are a very small subset of all vernal pools. So the ones in which this one is found are sort of the acidic versions of vernal pools. But this species can also be found in things that are not sort of what you would think of as a classic vernal pool either. So it can be found in things that we think of as vernal pools, but there are other bigger wetlands where they're also found. So I can't go into too much of the detail, but it's like, it's more complicated than that. So
Bob asks, how far outside the current known range have you looked for possible marginal populations? For example, have you looked at any sites in Androscoggin County?
I have not. I have not looked in Androscoggin County, and part of the reason is that window of opportunity that you really want to be in these wetlands or the accompanying uplands is so short, you know, that we are really trying to hit the sites that we think offers the best potential for them. But if we had a cadre of trained volunteers who are out at the right time of year, like mostly looking for an adult, frankly. I mean, obviously, if you have enough knowledge to tell a Williamsonia exuvia from another exuvia, great. But if you just focus on the adults and you come across one of these, especially the ring Boghaunter, in the uplands, I mean, that would be a huge lead. It's certainly, I would say it's possible. You know, it's certainly possible. But no, I haven't spent a lot of time looking outside of the area that we are focused in, which is basically York, Cumberland, and southern Oxford County. So
Great.
there's plenty of wetlands there to check out, you know, that we're still trying to identify populations.
Yeah, sure.
So,
All right. Laura asks, which water quality issues are most problematic to lintneri? Lintneri?
lintneri, yeah, lintneri. Yeah, I mean, maybe lintneri is the right pronunciation.
I'm not sure.
Yeah.
Sediments, chemistry, etc.
Yeah, my sediments. Yeah, I think I don't know that we know what is the most harmful. I think all of the above are potential concerns. Both, I mean, sedimentation is certainly a potential problem, I think, for these things. Because even though these wetlands are acidic, I would say they look relatively clean, thinking of all the sites that I'm aware of.
Mhm.
You know, the water can be quite tannic, but they don't look like they don't have algal growth of any sort, you know. So it could be sediment, it could be sort of phosphates or other nutrient loading that might occur in them that could really disrupt the kind of habitats in which we know that they occur. And I know that I don't think a ton of work has been done on that. Somebody did a study in Rhode Island looking at upland habitat use and potential development impacts, but it didn't get that detailed in terms of narrowing down that much.
Mm.
So,
Thank you.
sure.
Hanson asks, do ebony and ringed bog hunters compete? Do they threaten each other? And if so, which is more vulnerable to the other? Which I know we kind of talked about, but I don't know if you have anything more to add.
Yeah, they like I said, they can occupy the same habitats. So potentially in those habitats where they do sort of overlap in terms of breeding location, they there's the potential that they compete with one another.
Mhm.
And one time, I have to admit, one of our reference sites that I alluded to earlier, actually the first site that was identified in the state in South Berwick, I one time saw I forget exactly which it was. It was a male, I'm going to say it was a male ringed bog hunter that had clasped the neck of a female ebony boghaunter, or vice versa. I can't remember which one, but it was the two different species. They weren't mating, but they were in the beginning of what would potentially be a mating posture. I didn't stick around long enough to see whether it ever got that far. I don't think they probably would have been successful even if they had, because of the different genital characteristics and stuff. But it was interesting to see. It was like one of those really cool observations that you get to see when you're out there. I mean, I know there's more questions and I'm happy to take them. But one of the things I meant to mention and I didn't get a chance to is that it is such a joy to be out in these places at that time of year when an emerging tenorel is coming up out of its exuvial case that it's shedding. I mean, that is one of the most extraordinary experiences that I think a human can have. And I have been fortunate to witness that many times. And I never walk away feeling like, anything but the greatest joy at experiencing that. So
chills. That was great.
Yeah, I meant to include that in the talk, and I
love that.
forgot.
That's
So
great.
yeah. Get so caught up in the data and all this stuff, it's like you forget like the real part, which is to be there while it's happening. So.
I love that. Kathy Claire says field trip.
Field trip.
Charlene.
Yeah.
Charlene says they are tagging monarchs. Do you think those tags might work?
Too heavy. Those tags are too heavy. These things, their weight is like 0.1 gram. They like weigh hardly anything, the adults. And we looked at these like harmonic, these little things that would reflect a signal that are a lot smaller, a lot less weighty than the tags on a monarch. And this is several years back, and even that was going to be it would not allow the thing to fly in its regular way. It was like too heavy. So, but at some point, the technology probably will get to the point where we can do something like that with these things. We're not there yet, as far as I know.
Yeah, that's why Hansen also chimed in that wondering if microchipping or nanotechnology might get us there too.
I think so, probably so. Yeah, potentially we might be able to do something with like genetic material on the exuvia as well. We did a little bit, we didn't, but some folks that we collaborated with tried some stuff on the exuvia, relatively unsuccessfully. So we were unable to really and it destroys the exuvia. So we've been trying to not do that to these, especially the associated exuvia. They're sort of precious specimens that are stored at the State Museum now. So.
Oh, cool, great.
Yeah.
All right, let's see here. Nathaniel says that they'll be certainly looking out for it in Vermont this summer.
Woohoo!
Jason says, how frequently do you visit your reference wetlands before finding the first emergence of the year? Do you visit sites multiple times over the flight period? And if so, have you estimated detection rates? And again, these are as the talk went through, so I apologize if you kind of answered that already.
No, I don't think I did. I mean, it varies from year to year. Sometimes I try to get to the sites. My goal to both reference sites, I try to get there at least one time before emergence. Like one time when I think it's possible, but I don't think it's quite there yet. But I try to go to be able to confirm that they're not out, so that when I go, when I think that they might actually be out the next time, hopefully I'll find them. Cool. And I've had pretty good success with that. Yeah, that's that. And sometimes I've had to go back like 4 times to the reference site until I actually see emergence or adults in the surrounding uplands. And so sometimes those dates that I gave you, that sometimes they are actual emergence, and sometimes they're the first observed adult in nearby. So it's data sets are challenging that way. So
definitely.
Yeah.
Okay, we talked about the sphagnum already. What are your criteria to lump sites into metapopulations?
Yeah, it's sort of like proximity, I would say, you know, of wetlands. I mean, that one of those reference sites, there's like 6 wetlands in all within less, certainly less than a kilometer away. I mean, and they're probably all within 500 meters of one another, maybe a little bit more than that, but they're all within a kilometer of one another. And we find the adults, and we've documented all the wetlands as being breeding locations. So those essentially are, I mean, they're sort of like one population is the way we look at it, and the little individual breeding sites are sort of the meta sites where breeding is occurring at that site.
Cool.
We have we have three populations that have more than one wetland associated with them, and all the rest, the other 9 are all, at least as far as we know, are isolated individual wetlands.
Hmm. Cool. In your opinion, what are some of the most fascinating aspects of the ringed bog hunter?
Oh boy.
Big
Well, I mean, I think it is it's elusive, and therefore it makes it mysterious. You know, it's just not that easy to find, even when you know what you're looking for and you go out there, you know, at the right time of year. Yeah, it's I think that's what makes it the most captivating to me. And then this whole challenge with the ebony bog hunter and how to reliably distinguish the exuvia from one another is also sort of a fascinating topic. So, I mean, it's just a cool critter. It's just like, you know, I'm gonna tell a little story that I don't know if exactly addresses this, but like I think it was maybe 2 years ago, I was out at one of those reference sites with where there were emerging exuvia. And I was sort of, now I hit it at the right time. Maybe I'd been there once before, but I hit it on the day. I found them emerging. And sometimes when I'm at a site and I see emerging exuvia at that reference site, because I know there are other sites to get to, I'll be like, okay, here they are, documented, boom, move on to the next site, because I could find them somewhere else where I've never found them before. And for whatever reason on that day, I said, you know, I think it was whatever time of the day or whatever was going on. I said, you know, I'm just going to sort of like stick around until these 4 emerging individuals that I'm looking at actually fly, take their maiden flight, right? One of the most vulnerable times for them. Because they don't fly very strongly. Their wings are very, you know, they're just barely hard enough to even fly. They have a very fluttering flight. So I did that, and 2 of the 4, so these individuals that have, you know, so unusual, so uncommon, 2 of the 4 on their maiden flights, as I watched them, eaten by birds, like within, you know, within 100 meters of their flight. You know, somebody swoops in, munches them, gone. So I'm thinking, holy cow, you know, it's like there's so many aspects to this that even 20 years of sort of work around this species, and I still don't really have a clue what's going on with them, you know. So it's endlessly sort of intriguing as to what's really happening. You know, that of course sent me in all kinds of like thinking about like, oh wow, you know, there's elements. They were warblers. They were both warblers. I think one of them was a American redstart, and the other one was a black-throated blue warbler. And it was sort of late, and oftentimes I think when the first emergence for these species happens, those species might not be back yet. So I started to think like, wow, you know, the weird timing associated with climate change and things sort of coming at different time, you know, things, cycles getting thrown off. Is that something that is like a new threat, or a threat that was there that's enhanced because of climate change? I don't know. I'm speculative, but interesting, you know.
That's a great story, though. That's awesome and awful at the same time.
Awful, but yet nevertheless, it was like, I'm still glad I hung out to see it, even though I wished it hadn't happened.
Yeah, absolutely. Cool. I think that actually kind of hits the rest of the questions. I know I skipped over a couple. I tried to group some things that were really similar together. So if anyone here had any other burning question they wanted to ask, go for it. Otherwise, thank you so much, everybody.
Just want to say I found it in both of these books.
Oh, yeah. Yeah, good. It's in others. Yes. Yeah, Dragonfly through the binoculars. Yep.
And
Yep.
Mark, I was sort of curious when I was asking about a field sheet, maybe if we could just toss together like a really easy printout that I could put up on the MES website with this talk. That way, you know, if any of us are like, you know, like just going to go out to the field, we can just have something we can print out or pull up on our phone or something like that.
Yes. Yeah, maybe we can do that. That would be good. That would be really good. Yeah.
I'll remind you when I put this up.
The chat, I didn't even talk about it, but there's these other species that come out early. Those are the ones you're not. I mean, you could mistake it with a ebony boghaunter, but like the early white faces and the epitheca, those are the species that you're more likely to confuse a ringed boghaunter with, because they have a dark abdomen and they have reddish marks on their abdomen, on most of the segments of their abdomen. So those are the things that are, I actually thought about it and I didn't quite get to it. So it would be good to include that information on the cheat sheet, because for somebody going out looking for an adult especially, you'd want to know what are the things at that time of the year that also fly early that you're most likely to confuse this with.
Okay, perfect.
So, yeah.
Great, cool. Well, any other questions? All right, I think we're good. Thank you so much. That was really excellent.
Cool.
Awesome, Mark. Thanks.
Thank you,
Thanks.
Mark.
Well, thanks everybody for sticking around till the bitter end here, and hopefully it wasn't too much information. So,
Well done.